The Institute of Molecular Genetics in Pavia is one of the CNR Institute, and we are interested in RNA processing. This is a subject that we study since many years. In particular, regarding the RNA processing, we know that each gene encodes for specific transcript, but the transcript can be then processed to generate different protein isoforms through a process that is called alternative splicing. So our main interest in this project is to understand whether the alternative splicing has an effect or it is influenced during the Alzheimer pathogenesis. So at first we established an in vitro cell system that can mimic the Alzheimer pathogenesis. We used a neuroblastoma cell line, and we induced the differentiation of this neuroblastoma cell line with the different growth factors. And we judged the best conditions by looking at the morphology of the neurons, mainly by looking at the length of the neurites and the branches of the neurites. In parallel, we did an analysis of the neuronal markers, so we have found the right conditions by culturing the neurons first 5 days in retinoic acid, and then for other 7 days in a media that contains the brain-derived neurotrophic factor, vitamin D3, neuregulin, and nerve growth factor. After the differentiation, the cells have been incubated with oligomers of amyloid beta. So the amyloid beta is first dissolved in monomers, and then we reconstitute the oligomers and the fibrils. But then we use only the oligomers, and we incubate the cells for a short period with the oligomers of the amyloid beta. And what we can observe is that after 6 hours, the presence of the oligomers induces a phosphorylation of a specific residue of the tau protein on tyrosine 18. During these phases, we can also observe, in fact, by making a 2D gel analysis, that the isoforms of tau protein are affected by the presence of the amyloid beta. In parallel, we also search for synthetic peptide that can inhibit the phosphorylation of the tau protein on tyrosine 18. So we have synthetic molecules, different synthetic molecules that we first analyze in vitro, in a chemical reaction, and then in vivo, and we can observe, in fact, by treating the cells, incubated the cells together with the amyloid beta and the inhibitors of this FIN kinase, we can inhibit the phosphorylation of tau. So as we started at the beginning, our major interest is the RNA processing. So we looked at the alternative splicing of some relevant RNA binding protein, and we could observe that here we have one example. The SRSF1 transcript is affected both during neuronal differentiation and also during the incubation with the amyloid beta protein. So at the moment we have isolated the RNA from these cells, which mimic the Alzheimer phenotype, and we are analyzing the RNA by RNA sequence, and the data are at the moment under investigation in the bioinformatics lab that is present in our laboratory, that is directed by Dottoressa Bione, and they also have recently developed a new tool that is called Corella Genes, which allow us to investigate the transcript in these cells.